A flower doesn't just look beautiful, it functions as a highly complex biological engine, precision engineered for parallel cellular manufacturing. This cross-section reveals 4 whorls. Outside are sterile components for protection and attraction, the calyx and corolla. Inside sits the fertile core, the male androcium, featuring filaments holding pollen manufacturing anthers, and the female gynocium, consisting of a receptive stigma, connecting style, and protected ovary. The process moves from the pre-fertilization assembly of gametes through pollination and the unique mechanism of double fertilization to the final maturation of the seed. To understand how a seed is ultimately provisioned, we must first dive into the microscopic factories where the male and female payloads are constructed in total isolation. We begin inside the male manufacturing center. A cross-section of a young anther reveals 4 distinct chambers. These are the microsporangia, or pollen sacs. Lining the inside of these sacs is the tapetum. The tapetum is a specialized layer of nutritive tissue that feeds the developing pollen. If the tapetum fails to deliver these nutrients, the entire reproductive sequence collapses before it even begins. Inside the sacs, diploid cells called pollen mother cells, or PMCs, initiate microsporogenesis. A single PMC undergoes meiosis to create a tetrad, a cluster of 4 distinct haploid microspores. This historical diagram documents microgametogenesis. Focusing specifically on panels D and E, you can trace how a single microspore transitions from having one centralized nucleus to undergoing a specific unequal cellular division. This hand-drawn schematic breaks down that unequal division. The microspore splits into 2 very different cells. The massive vegetative cell, also known as the tube cell, manages logistics and energy. Suspended inside its cytoplasm is the tiny generative cell, carrying the actual male gametes. In many plants, the pollen grain is shed at this point, the 2-celled stage. However, in other species, the generative cell divides one more time via mitosis before release, creating 2 separate sperm cells and resulting in a 3-celled pollen grain. The mature pollen grain is a self-contained, multicellular delivery vehicle engineered to survive the journey to another plant. Deep inside the female hardware of the gynecium sits the ovary. Hidden within its protective walls are the ovules. This micrograph shows a single, enlarged diploid cell forming near the opening of the ovule. This is the megaspore mother cell, or MMC, the starting point for female gamete production. Through a process called megasporogenesis, the MMC undergoes meiosis to form 4 haploid megaspores. 3 megaspores rapidly degenerate and die, leaving only 1 functional survivor to carry on the sequence. In the next phase, megagametogenesis, the single surviving megaspore undergoes 3 successive rounds of mitosis. These are free nuclear divisions. The nucleus splits into 2, then 4, and finally 8 distinct nuclei, all floating within a single shared boundary without cell walls forming. Only after the 8-nucleate stage is reached does cellularization begin. The nuclei migrate to specific positions, 3 to the top pole to form the antipodal cells, 2 remain suspended in the massive central cell, and the final 3 move to the bottom micropilar end. Those bottom 3 form the egg apparatus. This consists of a single central egg cell flanked by 2 synergid cells. The structural thickenings inside the synergids are the filiform apparatus, which will act as a chemical beacon later on. Because the central cell contains 2 polar nuclei, The mature female gametophyte's final architecture yields a specific count, 7 distinct cells, housing exactly 8 nuclei. The ovule is now fully armed, a specialized multicellular receiver waiting for the exact chemical signature of a compatible pollen grain. The male and female payloads are mature, but they are physically isolated. Bridging this gap requires pollination, relying on either non-living abiotic vectors like wind or living biotic vectors like animals. This comparison shows the two strategies. Anemophily, or wind pollination, relies on exposed stamens, large feathery stigmas, and massive quantities of light, dry pollen. Entomophily, or insect pollination, utilizes colorful petals, nectar rewards, and heavy, sticky pollen designed to cling to a pollinator's body. Once a pollen grain successfully lands on the stigma, a chemical lock-and-key recognition sequence initiates. If the proteins match, the pollen grain is activated. What follows is a physical breach. The massive vegetative cell rapidly elongates, tunneling a tube through the style to deliver the male gametes. The pollen tube is steered directly to the ovule's opening by the filiform apparatus inside the synergid cells, which secretes a precise chemical trail. Pollination is an active, tissue-destroying cellular invasion executed to deliver 2 specific payloads directly into the embryo sac. The invasion terminates when the pollen tube enters a synergid cell and ruptures, releasing its 2 haploid sperm cells directly into the embryo sac. The first fusion event is called syngamy. 1 haploid sperm cell merges with the egg cell. This restores the diploid state, creating the zygote, which will eventually become the plant embryo. The second sperm cell bypasses the egg entirely and fuses with the 2 polar nuclei located in the central cell. This creates a massive triploid structure called the primary endosperm nucleus, or PEN. This process is known as triple fusion. Because syngamy and triple fusion occur simultaneously within the same embryo sac, this phenomenon is classified as double fertilization. This dual fusion mechanism ensures that a food supply is produced only if fertilization has successfully occurred, preventing the plant from wasting energy on an unfertilized ovule. In the immediate aftermath of fertilization, the accessory cells, the synergids and the antipodals, rapidly degenerate. The embryo sac is stripped down to the diploid zygote and the triploid primary endosperm nucleus. Survival depends on strict timing. The pen must begin dividing to establish the nutrient-rich endosperm tissue before the zygote is permitted to start its own embryonic development. The triploid cell executes rapid free nuclear divisions, followed by cellularization. This transforms the central cell into a massive, densely packed reserve of starch, proteins, and lipids, ready to fuel the seedling. With the food supply secured, embryology begins. The zygote divides into a basal cell and a terminal cell, growing into a globular proembryo and eventually maturing into a heart-shaped embryo that fills the cavity. At the macro level, the hardened ovule becomes the seed, while the surrounding walls of the ovary swell to become the fruit. From the isolated creation of spores to vector-guided pollination and the dual fusion of double fertilization, angiosperm reproduction is a system built to provision and protect the next generation.